From Microbe TV, this is Immune, episode number 48, recorded on September 8th, 2021. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back.
From Durham, North Carolina, Steph Langle.
Hey, great to be here.
From Madison, New Jersey, Brianne Barker.
Hi, good to see you all.
And we have a guest today from the University of Pittsburgh, Tim Hand. Welcome to Immune.
Thanks for having me.
Our pleasure. We're going to talk about one of your recent papers here today, but I want to start out by finding out a little bit about you, where you were trained and educated and trained and so forth. Where did you start out?
So I grew up in Canada. I grew up in Toronto, and that's where I actually started my work on immunology. I was actually very fortunate. Maybe you don't think all of it. So actually, I was very fortunate to work in or to go to school at an excellent public high school, and actually had a high school teacher who told me I was hell bent on doing microbiology as a undergraduate. I was going to the University of Toronto, which is a very large school. I mean, a lot of Americans don't realize that it's a massive school. You can study anything you want there. And he took me aside. He said, no, there's been this huge discovery in immunology. And what he meant was toll-like receptors.
Oh, wow.
He's like, you have to do immunology. This is the field that's the future. And so literally based on that one piece of advice, I did a major at the University of Toronto in immunology, which is a rare thing for an undergrad. And I loved it. I loved it from the very beginning. I've loved it since. And then so from the University of Toronto, I went and did my PhD at Yale University, working on memory T cells with Susan Keck, who's now at the Salk Institute, but was an amazing mentor for me. And, I did not decrease my enthusiasm at all for immunology. But while I was a student in Sue's lab, we were working on the memory to lymphochoriomeningitis virus, which is a very common model in immunology, especially for memory CD8 T cells. For those of you, who are aficionados, this is the big one. But I became fascinated with the work at the time that was just starting to sort of peek out into the world. It was coming from Jeff Gordon and David Relman, amongst many, many others, where they were starting to use next-gen sequencers to capture the microbiome and learning that it was much more diverse and complex than we had previously thought, building on the pioneering work that ecological scientists had done before that, sort of people like Jill Banfield, working at pond water and mine water and things like that, right? So then when they had turned those things onto the human microbiome, I thought it was fascinating. So I really only looked for postdocs where we could combine T cells and the microbiome, and I was very fortunate to be able to do a postdoc with Yasmin Belkade at the NIH. When I interviewed with her and said what I was interested in, she just said, that's what we do.
It's like perfect lab.
Yeah, it was lab. Taking T cells and putting them and trying to figure out how they respond to the microbiome. And that really is where, that's where I've been since, right? That's what we focus on in the lab. And basically almost everything we do is focused on trying to understand, this fundamental question of how does the immune system deal with colonizing microbes, period, regardless of whether they're invasive and sort of what we would call as pathogens, or whether they're sort of more benign organisms that might be helping you digest your food. We're trying to understand how the immune system parses those events. And now that we're at the Children's Hospital, Pittsburgh, I should have said. So once I finished in Yasmin's lab, I moved here to the University of Pittsburgh. I got an opportunity through the Mellon Institute here to start my own laboratory. And since we've come here, we have been interested in questions that are important in terms of pediatric populations. So we are trying to understand, how does the immune system make that first handshake with a microbe,
right?
Regardless of what the ecological behavior of that microbe is, right? Understand how the immune system is going to parse all these different signals and end up in a state of health or in a state of ill health. And so that's really what the lab does, if you know, in a very broad sense. And obviously we have beyond that very specific projects within that.
Yeah, that's great. Have you been in contact? What an insightful comment from your high school teacher. Have you still stayed in touch with that person? Because what a success story, right?
His name is Keith Brown. I think he went on to do a PhD and left Canada. Wow. And I think he's now, last time I checked, he was a teacher at a fancy prep school in Switzerland.
Wow. So, he might like to know that you're running your own lab based on his
yeah. I mean, he was a really wonderful biology teacher. If you can imagine this, I'm going to date myself a little bit. This was high school in the 90s in Canada. And he was an amazing high school teacher. We were doing PCR, you know, we were doing restriction into nuclease reactions. We were doing, you know, all kinds of things. I think we never did that. But those were the two big things I remember was we did our own PCR and, you know, moving the tubes between the water baths and all that stuff and then getting a band. But we were quite excited to get the band, I can tell you.
Is there any science in your family? I'm wondering where the interest came from.
No, actually. There's no real science in my family. I mean, my grandfather was a obstetrician, but not a scientist. He was really, you know, focused on delivering those babies. You know,
the first microbiome, right?
Yeah, exactly.
That would be the handshake happening.
Yeah,
right
exactly. But no, I mean, there wasn't a lot of science in my family, aside from the fact that, you know, my parents were incredibly nurturing and, you know, were very willing to, you know, I was super into science from a very young age. I was always collecting bugs and obsessed with stars and, you know, reading books about bacteria and viruses from the time I could read, like 11 and 12, and they would buy me every kind of book I could read. So they didn't have they still don't have any idea really. I try as much as possible, but they don't really have an idea about what I do, but they're very proud of it. And they were very, you know, they were very much in favor of it the whole way along.
So one of your other titles is you're director of the University Notobiotic Core. Maybe you could explain what that is. I don't think we've talked about that on Immune yet.
Sure. Yeah, that's a very interesting set of things that we do. So this is actually a really old microbiology and immunology trick. I think they've been doing it for over 70 years now. But with the interest in the microbiome that started around, you know, the early 2000s, the re-interest in the microbiome, you know, a lot of different university centers now have these facilities. So what they are and the best way I try to describe them is for those of you, especially for those of you who are older like I am, if you remember back in the old days when I guess maybe bone marrow transplantation is not as common as it is today, you know, there were children born with severe combined immunodeficiency that would have to spend time behind plastic, you know, physically separated from the world because any germs that got onto those children could be lethal. So that's essentially what we're doing with these mice. So these are mice that many, many years ago were derived using, imagine with a huge amount of antibiotics, were re-derived into a situation where they had no bacteria, no viruses, no protists, no fungi anywhere on their body, skin, intestine, lung, anywhere. And we now propagate them in that way inside of bubbles. And so when I mean bubbles, I mean literally these are like 6 foot wide at times, 3 foot high bubbles. They have gloves that you reach
into. The gloves, yep.
Yeah, they have these huge gloves that you reach into. We, you know, depending on the size of the bubble, you know, you can have anywhere between, 30 all the way down to sort of 4 cages of mice. And so the mice, you know, they grow themselves. They do just fine. They're not great. I mean, they don't digest fiber at all. But wow. You can propagate them continuously this way, keeping them away from bacteria. Everything that goes into the isolators gets autoclaved or treated in some way to remain sterile. And the real cool thing about the mice, and as Stephanie knows, you can do this as well with pigs. There are germ-free or notobiotic rats. You can do this with a lot of smaller species. But what's really interesting about them is not the germ-free state, to my mind, it's the notobiotic state. Because you have this sort of blank slate animal, you can then put in the organisms that you think are going to be most interesting to you. And you can then answer Koch's postulates to any given phenotype, right? You can say, for a long time, people in the microbiome world have been associating different bugs with different phenotypes. And germ-free animals are one way to get at mechanism, right? So you can say like, I think bacteria X is making my animal run faster and longer at night, right? This is an actual experiment that I saw in my mind. And you can then transfer the microbiome, the bugs, just from the intestine of that mouse into a new mouse and see whether those mice are driven to run any faster or longer at night by delivery of just those bacteria. So I think it's there's sort of a really cool tool, and when used, carefully, they can be a really powerful tool as well.
Absolutely.
Are they also virus-free, Tim?
Pardon me?
Are they also virus-free?
Right. So, virus is not my specialty, but so I imagine these mice will definitely have their endogenous retroviruses, right? So we're not going to be able to clear them out of that. But yes, other than that, they're, as far as I know, they are completely virus-free, right? So they're not going to have any of the sort of SPF viruses that we keep at our facilities at all. And nothing that has to be propagated from mouse to mouse is present in these mice.
So when these mice were derived, they started with kind of your normal wild type mice. So these mice are sort of genetically similar to the other mice that we tend to work with. Is that correct?
That's right. So we in our facility, we are currently, we're still sort of in our infancy. We've been around for about 3 years, but one of those years was a pandemic year, so we didn't do a huge amount of work in that year. I'm not making any excuses. That's just how things were.
That's just, yep.
So we're right now just propagating B6 mice and BALB/C mice, and for a while we had a really unique animal, which was a germ-free mouse that had a gene knocked out of it called RAG1, which is the gene that's responsible for recombining all T cell and B cell receptors. So those mice have no T cells and B cells. So those are a really useful model. We're not propagating those anymore. So, but yeah, we're essentially, we're breeding the two most common models used in immunology, BALB/C, which is, white mouse. A lot of people use it. And the other one is the C57 black 6, which is just the black mouse. I don't know. These are the same mice pretty much you see in pet stores. These are the most common mice that you're using in research these days. One of the
I was gonna say that it's you just mentioned pet shop, which I think is also a really important concept. Is that in immunology now we've gone to both extremes, right? Because the notobiotic, the germ-free facility is really the one extreme where there's no microorganisms at all. And you haven't talked about some of the limitations, but there are limitations to the analysis of these mice, right? Then there's the other extreme where you have wild mice that you just find or have from a pet shop, and they're chock full of all kinds of different microbes, and understanding how having those microbes on board or not on board influences how immune responses are generated is a really interesting topic these days.
I completely agree. I mean, we haven't done any of those experiments. There's an amazing young researcher here at the University of Pittsburgh, Kevin Cole, he is more of a zoologist, much more than I am. And he's looking at transferring the microbiome from different kinds of rodent species. He works a little bit on Paromyscus, which are wild field mice, those really cute things, huge eyes, huge ears, and he has a separate facility. I mean, Pitt is very concerned about all of these things that you're talking about that are the wild organisms that are making all the difference in our experiments. But Pitt is super concerned, as they probably should be, about those breaking out in our facility and infecting the entire thing. So he has actually a separate facility. I'm looking out my window as if that matters to people. So I where his facility is. I know it's not a visual medium.
So we do actually have a lot of people who watch this on YouTube, so they can maybe figure out where your office is on a map, right? And then assume.
Yeah. So he's actually, he's physically separate from where I am now, and he's looking at those things now. We pushed a little bit to be able to get him to do that in our germ-free facility, but it was made very quickly clear to me that that was a non-starter to bring those kinds of bugs onto the Pitt, Maine School of Medicine campus. But I'm looking forward to seeing what Kevin can get done. He's a dynamic guy, and I think it'll be fascinating to see what he does.
Yeah, that's exciting. And the other thing about the notobiotic facility is then, of course, the equipment that you need to sequence or to do experiments. So could you talk a little bit about, what other aspects of a notobiotic facility are required? So not just the animals, the cages, but then you have probably some equipment as well.
Sure. I mean, the big thing for us and our facility here that has been unique, as I mentioned, everything that goes into the isolators has to be autoclaved. So when we built the facility here, we actually built it mirrored. So I have 2 rooms, both which are exactly the same, but they have 2 separate autoclaves. And so the reason we did this is that when things break down in these facilities, it's because the autoclave breaks and you don't realize it, and you contaminate a whole set of mice and it's always the same contamination, by the way. It's bacillus. It's always bacillus because they form spores. And if you're going to make it to an autoclave, it's going to be a spore forming bacteria. So that's the way we have it. So these autoclaves I didn't quite realize it until I saw it. They're immense. They're 7 feet tall. They're about 3.5 feet wide. You can easily walk into them. They're 6 or 8 feet deep. We autoclave, 8 or 10 of these cylinders, these big, round cylinders all at once. The cylinders themselves are like 3 feet long and they have a diameter of about 1.5 feet. So these are, and that's how we're autoclaving all the food and water that these animals are getting. So everything is going through this autoclave. Everything's getting heated way, way, way up and steam sterilized before we can get these cylinders into the mice. That's the big thing. But other than that, the technology is not that complex. Right? All it is you cook these big cylinders, you attach them to the bubble, you sterilize everything in the bubble. So it's like having an airlock, right? So this is actually the metaphor I use when we train new people, right? Is you have to imagine that each of these bubbles is a spaceship, and if they touch the outside air, everyone's going to die, right? So you have to have, everything has to be sterilized. And if you're going to attach something to it, you have to make not an airlock, but a germ lock. Right. So we're going to attach the cylinder, or we're going to attach 2 isolators together. We're going to actually, what they do is they actually fill up, or they expand the airlock by filling it up with chlorine, like a chlorine mist. Like they super fill it, and then they leave it for 45 minutes and kill everything. And then you can just reach in and move across. But the technology, is not that complex, when you really think about it.
I thought maybe that I didn't know if there was also BSL-2 work going on inside the facility, if there's any experimental work going on. But it sounds like it's just the animals, correct?
Yeah, we did animals. I mean, the facility is BSL-2 rated. We do BSL-2 experiments, and there are some specific requirements for that. I mean, I'm not going to get into the deep technical aspects, but there's all kinds of airflow pressure things that we
put in place.
But in terms of BSL-2 experiments, this is one of the most safe ways to do it, right? So the mice are in a cage with a top inside a bubble that you literally can't touch. And if the bubble breaks, the whole experiment's toast, and we just take everything down and it's over. Right. So I cannot imagine a way that could be more, compared to a biosafety hood, this is even safer, right? They're physically separate from your world behind a filter. It's a pretty safe way to do BSL-2 work, especially most of the stuff we're doing is oral fecal infections. And if you somehow get orally infected by a mouse that's inside a cage, inside of a bubble that you can only touch with rubber gloves, you're really trying to get infected by that bacteria or virus. That's not an accident.
Right. Well, thank you for sharing that. I think it's good. It's nice that our listeners can kind of get a visual of the type of experiments that you do and how important that is for, how important germ-free work is. So diving into your paper, if you want to introduce it, talk a little bit about environmental enteric dysfunction and what we're going to be talking about today.
Thank you. This has been something that really started as a postdoc. So this was something that Yasmin was very passionate about it. So I should mention my postdoc advisor was Yasmin Belkade. prominent immunologists, laboratory chief at the National Institutes of Health. Amazing immunologist. I don't know if she's been on, but if you haven't had her on, she's incredible.
No, we'd love to have her on. Do you want to put a good word in? I know she's
put in a good word for you guys, for sure. Does amazing things. So, Yasmin was always very, one of the big focuses in the lab is leishmania, which is, of course, a disease of primarily of low and middle income countries. And Yasmin had a big focus in her lab. To ask, it's not sufficient to only work on things, even though she's working in the National Institutes of Health, she really was interested in global health and things like that. And so we chatted a lot in the lab about malnutrition, right? So she runs a big microbiome lab. Diet has been a big focus in the lab. And so, we just chatted a lot about that. And we were very quickly struck by this paper that had been published by Brett Finley's lab right when I was around leaving Dr. Belkate's lab, which was Brett Finley's group, which published in Nature Communications, which describes one of the first animal models of this disease, environmental enteric dysfunction. And we were fascinated by this because this was a disease that we had been thinking about for a while in the lab, and it was a disease that you never see in Pittsburgh, for example, right? So I work at a children's hospital. I can tell you there will be 0 cases of environmental enteric dysfunction in our hospital this year. If you're living in a low-income country, though, this can be your daily issue, right? And so what the disease is it's a disease. It's a combination of a diet that it's not necessarily too low in calories, but it's perhaps too low in protein and too low in fat. So you're maybe a different way of saying it is the diet doesn't have enough animal protein and animal fat in it. Right. So these children will not, by measuring their caloric intake, you're not going to be thinking that these children aren't getting enough calories. I mean, they're eating, but they're maybe not getting the perfect right calories. And then the second problem, this is probably more significant, is that in these low-income countries in particular, sanitation sometimes is not what we would like. Flush toilets are few and far between, and they're definitely not getting water piped into their homes that's been treated. And so what you can get is scenarios where children and adults both, they get chronically infected with diseases that people in high-income countries would think of as nuisances they get when they travel. So things like enterotoxigenic E. coli, rotavirus is a huge one, almost the most common one, always the most common one. Cryptosporidium, which is a parasite infection that causes diarrhea. Shiga toxin expressing E. coli, that's a real problem, really big, serious issue. But they'll be sort of assaulted with these again and again and again. And that, in combination with the low protein, low fat diet, actually leads to a scenario which used to be called tropical enteropathy, but that was obviously a loaded way of describing it, and spoke to the fact, it was just biased and not great. So then we moved to environmental enteropathy, but even that, for whatever reasons, people didn't like. And so the name changed to environmental enteric dysfunction. Some of the doctors on if they're listening to this, might know this disease from medical school as tropical sprue, right? That's what it used to be called. And so how it essentially manifests is the child will essentially, it's most serious amongst children, but the child, it can also happen in adults, but it's harder to tell because adults don't grow, but children will grow too slowly, right? And so they end up shorter in stature than they would otherwise. And also there's evidence that their IQ or their brain development is not what it needs to be. And this is very serious, right? Because these things cannot necessarily be fixed later on, right? You have a window when you need to grow rapidly. And if you don't grow during that window, then you can end up essentially it's not the greatest term, but you can essentially end up with stunted growth, stunted development, right? And in some of these low-income countries, the most common correlate of long-term income or your ability to break out of the circumstances that led to you getting environmental enteric dysfunction are your stature, your height, and your IQ, right? So if your brain has not developed the way you need it to, perhaps you can imagine a scenario where you won't be put forward in school to go to high school or to finish even middle school. And then your lifelong earnings have been affected by things that happened to you when you were 1 to 3 years old.
Right.
So this was a disease we really wanted to understand more deeply so we could break this cycle, right? And come in and really make the change. And so the last thing that's been so that fascinated Yasmin and us, and why we got so excited when we saw this model by Beth Finley. So I did everything in the reverse direction, right? Should have told you about the disease first and then the mouse model. But the reason we got excited was that one of the other things that's characteristic of countries where EED is endemic is that oral vaccines tend to do very poorly in those places. So oral vaccines are vaccines that are taken, underneath the tongue, or they're taken actually gastrointestinally. And these are going to drive B cells and T cells into the intestine and hopefully other mucosal sites where you're going to get protection at that mucosal site. Right? So these are things that should work great for things like rotavirus, right? This is what you want to do. You want to make sure you've got B cells and T cells at the intestinal space. We would argue same for enterotoxigenic E. coli. But in countries where they have a lot of EED, those oral vaccines do very, very poorly.
Right. And one of the other things then, well, actually going back just clinically, do you see the EED more prominent in the 1 to 3 year-old range, or is it older children that are also impacted? Because I'm trying to match up like what window can you implement therapeutic strategies depending on
right. So my understanding is that, and I'm not a clinician, I certainly don't work with children who have and adults who have this disease, is that you can actually get this disease as an adult all the way along the spectrum, but it's most serious in children who are growing, right?
So the younger, yeah,
stage.
Right.
Yeah,
right. Okay.
Right. Whereas in adults, it can cause. I should explain the last issue. That so the disease is essentially a disease of absorption. So what happens when you have these chronic GI infections along with this low protein, low fat diet is your small intestine actually starts. So it's again not a visual. Maybe people watching on YouTube will understand. But
he's holding up his hands and his 5 fingers.
The physiology of the intestine is, your intestine is not just a long, smooth tube, right? It has these finger-like projections or invaginations that go out into the intestinal space. They're called villi, and they do a lot of different things, but for lack of a better description, they increase the surface area of your intestine. So what happens in EED is that those villi become shortened and flatter. So you're actually decreasing how much space you have to absorb your food, and you're not getting the food necessarily you absolutely need. And so you end up sort of with an exacerbated malnutrition. So this can happen in adults. Famously, there was a famous paper looking at it in Americans returning from the Peace Corps who got this disease, and they came back to New York. And it was actually a really interesting study done in the 70s. So they were able to get a set of, because it's hard to get samples from people who live in low income countries, right? So this was an interesting group of sort of teenagers to 20-year-olds who were in the Peace Corps who they could examine. And interestingly, it took them 2 years for their small intestine to recover, for the villi, the invaginations to get as long as they were before, as they should have been. And you have to imagine that they weren't getting reinfected. They were living in New York, right? So you have to imagine they had access. I would hope they have access to sanitation and to high fat, high protein foods. It was the 70s, so I bet they did. So this can happen all the way along, the developmental pathway, but it's sort of most important in children, and that's why we focus on kids.
Sure. So one of the triumphs of vaccinology, in my opinion, is the near eradication of poliovirus, which has been done with an oral replication competent vaccine. So is that not susceptible to EED?
Well, so I would argue, right, and I don't want to argue with experts on this, on virology and vaccinology. I agree with you fully that the vaccination with the Sabin vaccine, the oral polio vaccine, has been transformational. I mean, I got that vaccine when I was in Canada. I would argue, there's an argument to be made that it's taken a long time to eradicate polio in the rural parts of Pakistan, right, in India. that was one of the last holdouts for polio. These places traditionally have been the most common sites of EED, right? So could you argue then that the polio vaccine works great and probably works better in the United States and Canada and Europe than it does in rural Pakistan, and that's why it's taken so long. And there have been studies saying that the efficacy of the oral polio vaccine is substantially worse in low and middle income countries than it is in Europe and Canada, for example, where it was still being used until recent years.
Yeah, it did take a long time in India, and we never understood why. And I know that a lot of the issues were revolving around gut microbiome and virome differences from other parts of the world that might be impairing. But I think EED is a reasonable explanation as well.
And I think, this is not my work. I mean, we definitely confirmed this in our mouse model. This is really the work of others. Again, Jeff Gordon, Bill Petrie, Brett Finley. The microbiome in these children with EED is very severely significantly changed. And so I think you could wrap that into where those studies where you're saying, oh, it's the microbiome. Well, I would argue that EED might be one of the biggest causes of change in that microbiome, right? So they're sort of overlapping. Those circles on a Venn diagram, I would argue, are very heavily overlapping.
So it sounds like this mouse model might be the great way to be able to test some of those things.
Yeah, right. I was just going to say that's part of what you did in this paper, right? Was you looked at what are the changes in the microbiome with
that's absolutely right.
The induction of EED or just the change in diet.
Right, that's exactly what we looked at here. So, so getting back, one of the things we had noticed for a long time was that this disease, which you would like to study in patients, is going to be a hard thing. I mean, people are now doing that. I've seen now amazing papers where they're getting duodenal biopsies from patients suffering with the disease. I'm not going to be able to get that at the. We're working now, try to build the relationships in halfway across the world to be able to start to work, with patients, but we don't have that currently. So. And, when I went to meetings discussing this disease, it was clear that a simple mouse model would be something that would be helpful. So this is why we set out to build the model that we did. And so working from Brett Finley's group, their paper had been focused on the shifts in the microbiome that were associated with the mouse model of this disease. Their model works, but it was quite complex. And so we felt like we could simplify it a little bit by using similar diets, but by using an E. coli strain. So when you read their paper closely, all of the changes that they had in the microbiome, the biggest results seemed to be that they were allowing greater colonization of the mucus and epithelial layer of the small intestine with Enterobacteriaceae. Okay, so that's a really fancy word, but that's a family of bacteria that contains all of many of the household sort of, for lack of a better term, pathogenic bacteria that are going to be found in the small intestines. Things like E. coli, Klebsiella, Salmonella, Yersinia. So Salmonella, cause of diarrhea. Yersinia, this is the cause. One type of Yersinia is the cause of bubonic plague. These are the bacteria that are found in the Enterobacteriaceae group. So this is a group of organisms that, through many, many, many studies, dozens of studies, are traditionally associated with inflammation in the small intestine. So when you looked at Brett's paper, what was going on? One of the things. Sorry, I should not. It was a very comprehensive paper. I should not limit it to something. But one of the things that was going on was the amount of Enterobacteriaceae, E. coli, Enterobacter, Klebsiella that was able to colonize the mucus of the small intestine was increasing. So we thought, hey, maybe we can just give an E. coli that spontaneously colonizes this space and make it work a little bit simpler and easier. And that's what we did. And it took Amrita about, sorry, I should mention as well that all of the work we're going to discuss today was done by an amazing postdoc in my lab, Amrita Bhattacharjee. So it took Amrita about 6 months to get this up and working. But what she very quickly realized was that and we're still trying to figure out why this is, but that the low protein, low fat diet somehow changes the surrounding microbiome to allow E. coli to colonize the small intestine and the large and the colon at a much higher amount, which was exactly what had been shown in the previous paper and was sort of known to happen in the small intestines and colons of patients with AED. So that was very gratifying.
And a question related to that. So when you did the diet, it's low protein, low fat, but you have to supplement that with something else because they're isocaloric, right?
So that's a great question. So no. So we don't supplement that. So they're actually getting, I think it's about half the amount of fat and 1/3 of the amount of protein as a mouse normally would intake. And this is very, very important that we talk about this. If anybody's interested in doing the model, by the way, and if you're interested, if you're an immunologist or a microbiologist, please just get in touch with me. I'm easy to find. But this is an important thing to mention, was that we had to restrict their calories. And the reason why was when we first tried to do the model is that mice, much like humans, they detect satiety based on fat content. So if you give them a diet that has, 1/2 or 1/3 the amount of fat they should get, they eat 2 to 3 times as much as the diet. And so the mice on the malnourished diet were getting obese because they were eating three times the calories as they were supposed to in a week. Right. So they're supposed to get a certain amount of kilocalories, and they're eating 3 times that much. So what we had to do was we had to feed the mice a specific number of pellets each week. And it was, we worked everything out now, but it was, for a while, it was difficult with the animal facility because they were constantly calling us saying our malnourished mice were out of chow because they would eat it in 2 days when we'd given them 3 or 4 days worth of food, and they'd be empty. And we'd say, okay, no, they're fine. They've just gorged themselves.
Don't give them more.
Don't give them more, because they just never feel full. So that was incredibly important. We actually, we measured essentially calories based on a number of pellets. And so that was incredibly important for the model. So no, we weren't. So the both diets have the same number of calories, it's just where the calories are coming from.
And
so if go ahead.
No, you go ahead.
I was just going to say, if the calories are coming from somewhere else, are they coming from carbohydrates? Could that, so that then you're potentially feeding different amounts of carbon sources to the microbes in the gut, which could also account for shifts in the microbiome that then would allow those E. coli that you're giving them to colonize more easily?
Yes, I would agree with that entirely. So we are now trying to do this sort of heavy lifting on that and trying to understand in a complex microbiome, what does that diet shift mean in terms of not just which microbes are there, but which genes are being expressed by those microbes in terms of their digestion. And then how is that then affecting that mucus layer, right? Because that's the part where the sort of the rubber meets the road. That's the part where those bacteria are going to be interacting with the host. And that's where we most want to understand, so it's sort of a 3-way interaction. And I find difficult to comprehend. So I hope, I hope it's not too convoluted, but we're trying to understand exactly what you're saying, the interaction between the epithelium of the host, right? That's the cells that the host has made, the microbiome that's there, right? And what genes it's making and how it's affecting the host and the other members of the microbiome. And then finally, we're going to add in, an E. coli that naturally wants to swim into that mucus. And somehow we've changed the microbiome in such a way that now the E. coli is swimming more into that mucus and colonizing it much better.
And you're also eventually going to look at metabolites that the bacteria are making, because they also influence a lot of these outcomes as well, right?
Yes, we've done that. I mean, I think that was done beautifully, actually, in the Finley paper. We're trying to see if our model is different from theirs or exactly the same. But bile acids were very significantly different in the Finley paper. I would anticipate they're also going to be I'm I'm meeting with our collaborators who did that work on Friday, so I have no idea what the results are. It's sort of black. Someone knows what the results are. I just don't know what they are.
What are the signs that convinced you that your mice do have EED?
That's a great question. So the two things that convince us the three things the mice convince us are EED is, one is that they were stunted, right? This was critical to us. And so, and they weren't severely stunted, right? They were a little bit stunted. So these are not mice that don't grow. So we started the model on mice that were adolescents. These mice have just been weaned. It's very important. So 3-week-old mice. So they've just come away from their mother. And so these mice still grew, but they grew 50% less. So instead of growing 5 grams over the 4 weeks or 7 grams over the 4 weeks of the experiment that we were doing, they grew, 3.5 or 4 grams. So they're still growing. They're just growing a lot more slowly. That was an important thing. And also they ended up shorter, right? Their tails are shorter. They're shorter mice, but they're not so short that when you open the cage of the mice, you say, oh, these are tiny, right? They just look normal and they run around, they're fine, they seem fine, but they just are smaller a little bit. And it really only comes out when you carefully measure weights and tail lengths. So the last thing and most important thing, though, was that their ileums looked like the ileums of patients with EED, right? So the ileums had that flattening of the villi, so they were not going to be able to absorb as much. And finally, they also seemed to be invaded with a bunch of immune cells when we looked at them with a microscope. Right? There just seemed to be a lot more cells invading the lamina propria. That's the space just underneath. The lamina propria is the fancy word for the space in your intestine just underneath the epithelia. So that seemed to be invaded with a bunch of what were pretty obviously immune cells when we looked at it under a microscope.
And so then now that you had this model, your EED model, which was dependent on both the low fat and the low protein diet, and then the attaching E. coli species, you're then going to go on and test and say, okay, is this phenotype, can we repress vaccine-induced responses? And you used a double mutant lymphotoxin.
That's right.
Correct? Okay.
Label toxin.
Yes, label toxin. Thank you. And so in those experiments, what were some of the things that you found? And specifically, it was really interesting. The intestine is not a monolith. You found different responses depending on what the different parts of the intestine.
Sure. So so the model we sort of used for our oral vaccine was a model that I've been using for a few years. I started using it when I was a postdoc in Yasmin's lab, and it's a long-term collaboration between my lab and an oral vaccine lab at Tulane University that's run by Betsy Norton. And Betsy makes this double mutant label toxin that is actually in clinical trials right now in humans as an adjuvant for an oral vaccine. This is an incredibly potent vaccine in people and as well in mice. And that's why we use it, because we wanted to use something sort of very big. So I should mention as well that this is the causative agent in diarrhea and traveler's diarrhea, right? So this is, if you've been unfortunately ever had traveler's diarrhea, this is what caused you to have diarrhea. But for us, it's great. It induces a massive T cell response in the small intestine and a massive IgA response. And when we gave it to mice, we saw the same thing. And we were fortunate enough to do a trick that I've done a few times now. We're able to identify the dominant antigens. I guess probably your audience is more familiar with antigens now than they ever have been because of SARS-CoV-2. Yeah. We were able to identify the dominant antigens in this toxin. And it was easy because the toxin is a protein, right? So it has T cell and B cell epitopes in it, and they're easy to find. There's algorithms, and we were able to figure it out. And using those peptides, we're able to very carefully isolate and analyze the T cells that are responding only to the vaccine, right? So we can, in the gamish and the milieu of T cells and B cells that are all through your intestine, we can very precisely follow only those T cells that are responding just to the vaccine. And so with these tools, which is the double mutant label toxin, so what that is the 2 mutations that have been made in the toxin mean that the mice don't get diarrhea, right? So even though if you drank label toxin, you'd get horrible diarrhea, and this has been shown actually in a number of studies, this double mutant label toxin doesn't cause any diarrhea. Okay, so the mice really don't see any negative side effects. They don't lose weight. They seem fine. It's an oral vaccine. But with these tools, what we could then do is we could then apply this into our EED model and measure in our model, are we actually seeing that oral vaccine failure that was typical of places where EED is endemic? And indeed, we saw that failure very specifically. And what was so fascinating about the failure, which Stephanie mentioned, was that it was happening only in the intestine, right? So the intestinal T cell response was failing. The intestinal antibody response seemed to be very significantly diminished. And we did this by measuring immunoglobulin A that got secreted out into the intestine to try to deal, right? Your immune system thinks there's a bacteria there, so it's secreting a bunch of antibodies out to deal with that vaccine. But in the mesenteric lymph node, which are the lymph nodes that drain the intestine, we didn't see any difference at all. So it seemed like the T cells and B cells are being primed, but they're struggling to accumulate at the mucosal tissue where they're going to be most necessary.
That's fascinating, because looking at the paper, based on the numbers in the lymph node, they weren't being retained there. I'm trying to think, is the difference because the trafficking signals are lower? Did you look at maybe some of those like alpha 4 beta 7 or trafficking chemokines to say that maybe they just aren't migrating back to the intestine after they're being primed?
Sure, that was one of the first things that we thought of. And so the problem though with doing those experiments was it's really sometimes hard to do experiments on cells that are absent, right? So we only had the cells in the lymph node. We didn't have the cells in the intestine. Most experiments, there were no cells to test in the intestine. So that did make it hard. But when we looked in the lymph node, we shockingly didn't see a difference in CCR9 and alpha 4 beta 7 expression. So it seemed like the cells were being primed correctly. And to get deep into the sort of weeds of it, I had previously worked on a project as a postdoc looking at long-term damage to lymph nodes and lymphatics that can be caused by oral infections with things called like Yersinia. And then in case we were using Yersinia pseudotuberculosis, which is an organism that causes damage after an oral infection to the lymph node in particular. You get a big, huge permanent abscess on that lymph node. And in those circumstances, we saw the same thing, but there it was the priming of the T cells in the lymph node that had been broken because the lymphatic system wasn't working correctly. Here, the dendritic cells were trafficking just fine. We tested that pretty carefully. And it looked like the T cells were getting the right address signals so they could end up in the intestine. So what we hypothesized at that point was there's something local about the small intestine that was preventing the cells from functioning once they got there. And so This is, again, we're going to go a bit deep in the weeds here, but one of the things that has been noticed in work by Dan Littman, amongst others, as well as Yasmin Belkade's work in the skin, is that T cells that are going to travel to these barrier tissue sites, they're not done differentiating and proliferating in the lymph node. They have to receive extra signals once they arrive at their tissue, endpoint. And that's what we got interested are those signals that are necessary for the further differentiation and proliferation in the small intestine, that's what's being affected. And so that's when we started to look much more deeply at what types of immune cells are actually invading that small intestine, right? We looked at the small intestine with the microscope and we noticed that there was an increase in immune cells there. And so at this point, we really wanted to go deeper into what the differences were between the small intestine with and without the experimental EED where we were causing these mice. And that's when we discovered that there was a massive increase in regulatory T cells. I shouldn't say massive, I'm exaggerating. It was about twofold increase in regulatory T cells, but only in the small intestine, right? So really, again, a difference that was only confined to the small intestine, which is where the disease is largely happening.
So it's not as if maybe the cells got lost. It's that the cells changed into the wrong thing when they got to the location.
Right. So one of the things, yes. So what we think is happening is they're getting to the intestine, but they're not then doing what they need to do to be effective once they get there. They're not proliferating. They're not differentiating. And there's just sort of, the response is sort of petering out.
And using, go ahead, Cindy.
Oh, I was just going to say the regulatory T cells that you see there are not the ones I typically expected to see.
Right. So, this is one of the things that this is one of the specialties of the lab is phenotyping T cells in the intestine. If we're known for anything, that's what we do. So we pretty quickly discovered that the T cells were co-expressing a transcription factor, ROR gamma T. This is the transcription factor that has traditionally been associated with IL-17 production and IL-17 producing T cells in the skin, in the gut, wherever you find them. This is that transcription factor. In the last 5 years, though, people have noticed, actually way before that. I mean, there's actually a very old paper from Dan Littman and Sasha Rudensky that describes these cells many, many years ago in the intestine. It's been reinvigorated by people like Diane Mathis and Gerardo Beryl, finding these cells everywhere, all over the body. I mean, Diane has done really incredible work in that area. Most of that had been focused on the colon. Here we were finding regulatory T cells that co-express this ROR gamma T transcription factor in the small intestine. And that was an important discovery because, in our first experiments, when we discovered that Tregs were increased, we of course used this fancy trick that had been developed where you can drive a diphtheria toxin receptor downstream of the FOXP3 transcription factor, which is the defining transcription factor that induces regulatory T cells. And so if you then inject those mice with diphtheria toxin, you deplete all the Tregs. And when we did that, we were able to completely restore the oral vaccine response. So we knew Tregs in general were important, but that mouse is a bit of a heavy hammer, right? Removing all the Tregs, of course, you're gonna increase immune responses everywhere. So restoring the intestinal immune response was not that exciting. Although Amrita was very excited by it, but I was a little nonplussed. But Amrita is a very, very engaged postdoc, and she already had a new trick up her sleeve, and so she was already breeding at the time an animal where we could inducibly, using all the fancy immunology genetic tricks. She had an animal where we could inducibly remove just ROR gamma T expression. The gene that drives it is called RORC. So we were able to just get rid of that gene, but only in T regulatory cells. And when Amrita did that, she saw that it phenocopied the total Treg reduction, and now oral vaccine was also restored just by getting rid of that one subset of T regulatory cells. So that was pretty gratifying.
Yeah, good thing she was breeding those animals, right?
Yeah, I mean, it wasn't that much of an accident, right? Once we had the Treg result, we knew we could get the FOXP3 DTR mice very quickly from John Williams, who's actually my boss here at Children's Hospital. And then we were very fortunate to be in the same university with Dario Vignali, who was able to give us the inducible FOXP3 Cree mice, and we just had to breed them to the RORC floxed animals, and it took us 6 months, but it was very, very much worth it. But yeah, we had to get those started pretty quickly.
And so once you found, is there something particular about the ROR gamma Tregs? They seem to really be heavy handers in the suppression of immune responses at the site. So is there previous evidence of those particular cells really dominating the suppressive response of Tregs?
I think in the intestine, certainly, that seems to be the case, that they seem to be particularly critical for dominating these responses, and in particular for scenarios where inflammation is being driven by the microbiome, right? So, and I keep on name dropping the Littman lab, but his lab has been very important in my field. So his lab showed that. For Helicobacter, which is a colonic bacteria that drives colitis, that ROR gamma T expressing Tregs were important for mediating that. Diane Mathis has shown, I don't know, in like 5 models now that these ROR gamma T expressing regulatory T cells are particularly important for maintaining quiescence with regards to the colonic microbiome. And Gerardo Beryl showed that they were important actually longer than that, so that these, at least in mouse models, these these ROR-gamma T expressing Tregs may be important for the long-term reduction of inflammation at many different barrier sites and things like asthma and allergy as well.
Yeah. So that was going to be my question, was that I'm really interested in sort of the tissue specificity here. So how much of this, particularly with these gamma T cells, is specific to the intestine? Do we see this in other mucosal tissues? And do we see this in other barrier organs? And so how can we think about this with some other types of immunologic diseases?
Sure. Definitely they're very prominent in the colon. That's where they've been seen in the largest numbers at steady state. So in that work, they seem to be very responsive to signals from bile acids that are the small amount of bile acids that are escaping the ileum. Those seem to be important. And in particular, bile acids that have been modified by the microbiome are critical for their function and their induction in the colon. ROR-gamma Tregs have also been seen in the lungs and in the skin, right? And it's, in each case, I do think that there are critical regulatory cell subset. I don't really know if I've put it together in the way you want me to, in terms of trying to get like a deeper, synthesis of what's going on. My reading of the literature would be that they seem to be particularly important or responsive to situations where you're getting an irritant. If I don't want to personify these bacteria or parasites or fungi, but where you're getting an irritant, right? Something that's non-invasive, but is attaching, right? And is inducing an immune response. That seems where they seem to be most important. And that's what I would argue their utility is and why we're seeing them so much, because these are the sort of organisms now we're using to modify microbiomes, right? So we're less so going right after them in terms of using something like Yersinia, which I used to use, right? Which is a pretty aggressive organism, right? It's not, it's seeking out Peyer's patches, it's invading, it's causing a massive infection inside the host, right? Whereas ROR-gamma, you're not going to see a lot of ROR-gamma Tregs there, right? There's no point in regulating the immune response against Yersinia. You just need to protect yourself against Yersinia. It's in that gray area, right? These organisms that sort of attach to you, but aren't necessarily going to invade your lymph nodes and your blood and kill you. That's where these Tregs seem to be important. And if you'll allow me, this is the segue. I think that would help me to explain, like, actually what we saw was there was actually a bit of a double-edged sword when we removed those ROR-gamma Tregs, which is that the mice that had EED that had failed the oral vaccine, indeed their vaccine responses came back, but now they actually lost weight. Mm-hmm. Right? So now their stunting became incredibly severe. Mm-hmm. And what we argue and we hypothesize we're now looking at more closely is that what these ROR-gamma Tregs are doing is, we've evolved to have these Tregs in our barrier surfaces to shut down chronic inflammation at those sites under extreme circumstances, things like EED, so that you can tamp down inflammation, but you can maintain systemic immunity, right? So, we're keeping the host from dying from chronic GI inflammation while allowing the systemic immunity that's going to be driven by the lymph node and the spleen to remain intact, right? So whenever we did a systemic infection in EED mice, including the oral vaccine, they were fine.
Right.
They made normal responses. It was only the responses at the mucosa. So, we're arguing, and this has been clearly shown by others in the literature, that the immunity is compartmentalized. Right. So you can shut down immunity in your small intestine, but leave everything else on in a hope that you're able to sort of reduce that inflammation, get it under control, but still maintain vigilance against the possibility that those organisms that were causing the problem in the small intestine are actually Yersinia and they're going to kill you. Right. And I've personified two things that don't think, which is the immune system and bacteria, but it's the way I think about it, and I think it's helps.
Helpful. Yeah, no, I think that was really helpful, because sort of what I was starting to think about were the ways that vaccines potentially delivered systemically or to other mucosal sites might be modified here. And what I get from your answer is that this is also very related to the type of organism and some of the features of, say, the attachment of the type of organism, which I never would have thought of. So that's super fascinating.
Yeah. I mean, I think we need more evidence, but the evidence seems to be, that these regulatory T cells are responding to organisms that are attaching or spending a lot of time in the inner mucus layer. I'm very gut focused in my lab, so I apologize. I don't know anything nearly enough. I don't know enough about the lungs and the mouth and the skin, but at least in the intestine, that's what we're seeing. So those bugs that spend time in that inner mucus layer, that sort of thickest mucus of your intestine, those are the bacteria that are the most potent inducers of these regulatory T cells.
I'm going to be the non-immunology listener who wants to understand the big picture here. So first, two things. What are the regulatory T cells doing to blunt the vaccine responses?
So that's a great question. We're working on that now. We actually don't fully know. So we in others' work, they've noticed that these T cells are very potent producers of the cytokine interleukin-10. That could definitely be part of the story, although we didn't measure huge increases in interleukin-10, but we're definitely following that up now. The thing we have noticed that was very fascinating is that the regulatory T cells that are making RR gamma T seem to express higher levels of two surface molecules. And I'm sorry, immunology nomenclature is terrible, but they're called CD39 and CD73, which don't mean anything to you, but these are molecules on the surface of cells that cleave ATP and ADP down to adenosine. And adenosine around immune cells is very anti-inflammatory. So that's what we think it might be important here and might be meeting a lot of the effects. So we're changing the amount of extracellular ATP and extracellular adenosine, changing that balance, I think could be an important part of the story, both for the epithelium and also for the other immune cells that are around it. So those are the two things we're following up currently.
All right. The other part is the regulatory T cells, are they constitutively present as a consequence of EED, or are they induced by the antigen that you put in?
Right, so you've gotten both of the big unanswered questions. Thank you. So these RR gamma T expressing T cells are present in the small intestine at low levels, but they're very, very substantially increased by EED. We have long hypothesized and are trying still to prove that they are specifically responding to the E. coli, but have not yet been able to prove or disprove that, which is an absence of evidence argument, which is the worst argument. So I wish I could tell you that we knew that they were particularly responsive to the E. coli. What we know is that the E. coli has to be present, right? And then it has to be an inherent E. coli to get them to arise. So if you just put in an E. coli that, for example, the E. coli we put in an E. coli that can't swim, has no flagella, doesn't get into the mucus, and that did not induce these Tregs. But we've not been able to do the killer experiment I think that you want. I'm sorry about that.
Another big picture question regarding antibiotics. So one of the experiments that you did when you gave the mice antibiotics, correct me if I'm wrong, but it did relieve the vaccine suppressive effects of EED. And so one would then argue, well, can you give antibiotics to children prior to giving a rotavirus vaccine to have an immune response? But then, of course, the consequence is increasing intestinal inflammation that would lead to further stunting. So could you talk about that and how you see antibiotics playing a role?
So first, I mean, again, not a physician, so you should not take my medical advice about the use of antibiotics. You should follow your doctor's advice. But I do think that there could be a case to be made that because the microbiome it seems to be so important in this disease, and you need to reset it so that you can fix this situation in children and mice. I would argue that maybe that should be something we should be going. So use antibiotics temporarily, reset the microbiome then by reestablishing the diet and getting away from the scenario where you're getting chronic infection with these inflammatory E. coli. That might be a successful way to deal with the disease. Although I will mention, this is a detail from the paper, but something which we've noticed in my work and both of the papers that in my work that we've done on this subject is that when you deplete the microbiome completely, the whole level of the vaccine response is reduced. So when we treated with antibiotics, indeed, we saw that the mice with EED had the same vaccine response to the mice that never had EED. But I should mention that response was significantly reduced compared to just a regular old mouse off
Right.
cage, right? The microbiome is important for oral vaccines, right? You have to have, for lack of a better word, the right microbiome.
Right. And that's what one of the things we saw in pigs as well is when you gave antibiotics, it decreased the response to both challenge with rotavirus and the vaccine as well. So it's very complicated. And thinking about, these functional foods. And so Jeff Gordon had published, I think, the first evidence in a clinical trial of giving malnourished children microbiota. I forget which one it was, and it improved growth and development. So are you working with any thinking about using microbes then to treat EED?
We are not currently looking at that in terms of using functional foods and microbiome. I think that research is fascinating. We've not looked at that currently. We're currently more focused in our lab in looking at what are the components of the microbiome that drive the disease.
Right.
I think you have you have to sort of pick your poison. Yeah. I think that Jeff Gordon and Justin Sonnenberg and people like that have that sort of area well covered.
Yeah, sure.
And, I think that their work is fascinating. I'm very interested to see what they see. And I would, in the future, I would definitely be open to looking at those things to see whether they can fix the system. But I think the take home from their work is the same as mine, which is I don't think it's going to be sufficient to just fix the diet if the diet's not fixing the microbiome as well.
Right.
You can't just give back fat and protein. And I think the take home from a lot of that work has been the functional foods. The food has to actually fix the microbiome, otherwise you're not going to get restoration in the child or in the patient.
Is it useful to now look in people and see if there are increased numbers of Tregs in the intestine, people with EED?
I imagine that will be done now. I mean, I think, but in the few studies that have been done on the duodenums of children, I think it was done. I know it was done on children in West Africa. They discovered that there was a substantial increase in T cells in the duodenum expressing CD3 and CD25, which is the exact molecules you would expect for T regulatory cells. They didn't at the time look for the killer, smoking gun, which would have been to look for the transcription factor FOXP3. They just didn't look at that in that study. But I anticipate those studies are coming.
So I'm sort of interested in the timing aspect of this as well. I have the impression that, for example, the children with EED may not have EED their whole life, or at least some of these patients with EED have it sort of for a short period of time. And so what happens with the regulatory T cells as the disease resolves? Are they going away? Are they going somewhere else? What do you kind of have as your model there?
Yeah, I mean, that's a great question. And that's something, I mean, they've all been great questions. But this one hits particularly on my long-term interest in memory cells. So we've been fascinated with the ideas. If you guys will let me go deep into this a little bit, we've been fascinated.
We like
with
the weeds here.
Oh yeah. Are there memory responses against non-invasive bacteria that are in your intestine? Like, do you actually make memory CD4 and CD8 T cell responses against the bacteria in your intestine that stay in your intestine and don't invade the body? We know if you have an oral infection with like your Listeria, Yersinia, they make perfectly normal, powerful protective memory responses. Unclear for non-invasive organisms, right? And so perhaps unsurprisingly, when we've looked at mice that are recovering in the mouse model of EED, we see the R gamma Tregs, they go away pretty quickly. Like within a few weeks, they've restored themselves back down to what, quote unquote normal numbers. What we're looking at now, because the disease in many children is quite chronic, and our model was very short, only 4 weeks. We're now trying to build a model where the mice will go through this process over 12 weeks. Right, and then see, are the pathways then much more permanent, and then is there a much longer period to health? Because as I mentioned in this seminal study done in the 70s on Peace Corps workers, when they came back, they obviously didn't look at regulatory T cells back in 1973 because we didn't know that they existed. But the health of their intestines, and this was being done by an absorption study, took 2 years to be restored. So we would anticipate then that, if you got really chronic EED, if our model really models human D in any way, that these kinds of changes would be much more persistent. But we just don't know that yet.
In that timing question, it goes back to an earlier question, and I'm sorry I didn't clarify. I was curious, not so much about children versus adults, but more, the different ages of children. So let's say if you have EED as a 1 to 3 year old, or if you had maybe a normal gut development, but then you are a refugee and then you're going into a displacement camp where you don't have appropriate nutrition as a teenager or someone going through puberty where you're growing. But of course, this is a much different scenario. Do you have any thoughts on what happens if you have the normal development in that window, but then you get EED maybe a little bit later when you're still growing?
This is now I'm going way out of my depth.
That's fine.
Yeah, I just don't know the differences about bone growth and brain development between smaller children and adults, and how they're affected by fat and protein concentrations in the blood. I imagine it's quite complex.
Right.
Unfortunately, I think I'm just way out of my depth on that question.
I think in a mouse model, you could maybe do that, right? Yeah. You could disrupt it and then try to fix it later on.
Yes, that is something we should definitely do. That's a good idea. We have not done that, but I will mention that maybe in this case, the most, perhaps argue that in not all cases, there are mouse models the best models for humans, right? So the mice have a much shortened development, time frame. And even I don't even think you can really extrapolate it that well to human adolescence, right? I mean, humans they're just developing for so much longer, not just in terms of time, but also in terms of how long in each stage and different things. I think it's a great idea. And I think, if we have the chronic model, going on a month, off a month, on a month might be able to do what you're asking, Stephanie. That's an interesting possibility. But also it may just be that mice here are not a great model for that kind of thing. We'll see.
Yeah, sure. Thanks. Well, great. Does anybody have any kind of more questions? Or Tim, do you have anything else you want to say about this great paper? And congrats to your postdoc for all her work.
No, I just want to say thank you to the people, who helped us out on this work. All the people I started this as a postdoc in Yasmin Balkaid's lab. So
wow.
Great. Yeah. The initial work from this was done when I was a postdoc almost 6 years ago now. So it was a long road. And then Amrita has been a trooper, and just say thank you as well to Betsy Norton, who's been a real long-term collaborator of ours and continues to be a steadfast, scientific friend in all of this. So, and thank you guys for spending the time with me. I'm sorry to go long if I went long.
No problem at all.
You're good.
No, it's great.
We like long.
Chatting about this topic. I really appreciate the interest and, you guys spending the time with me. It was a great chat.
We like long and we like details.
Yeah, that's one of the things we're trying to do on all these pods, is to prove that people can handle long conversations, not just 5-second sound bites, right? Which I think confuse issues more than sort them out, unfortunately.
Yeah, I hope I gave you a picture of the subtlety.
Yeah, it's lovely.
But this is an animal model, right? So
yeah, sure.
I think we can extrapolate some really interesting things, but I will be the first to mention that I think we need more work in the actual people who are affected with this horrible disease. And I'm very much in favor of that.
Yeah. No, animal. We like to say over on TWiV, mice lie and monkeys exaggerate. They're all models, and you have to remember that. All right. So that's immune number 48. You can find the show notes at microbe.tv/immune. If you have any questions or comments, please send them to immune@microbe.tv. And if you enjoy what we do here on Immune and all the other podcasts where we try and dive into science deeply, where you're not going to get on CNN or Fox News, you're just not getting it there, folks. Here's where it is, and we don't charge you for it. So if you'd like to support that effort, microbe.tv/contribute. Our guest today from the University of Pittsburgh, Tim Hand. Thank you so much for joining us.
For having me, everyone.
Cindy Leifers at Cornell University on Twitter, Cindy Leifer. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University, Stephanie Langle on Twitter. Thank you, Steph.
Yeah, thanks. This was fun.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. This is great. I learned a lot.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.